Hey there, we're back, and we are still talking about fluids, electrolytes, and acid-base balance, and we are going to go into electrolytes right now and talk about electrolyte balance a little bit. Now, there's a lot of detail in your book about this, and we are going to hit some of the basics of it in the lecture notes. Some of these things you've covered already, especially some of the ones like sodium and a little bit calcium, potassium. So some of this will be review, and then other things will be new. But the detail level that's in the lecture notes is about where I want to be. However, if you want more information about any of them, reading the book in a little more detail is a great way to do it. So what are the functions of electrolytes? And again, this should be mostly review. We've probably covered all of these things so far. And one is that they provide essential minerals. The second is that they control osmotic fluid movement between compartments, and especially when we think of fluid movement, we think of sodium, and then to a lesser extent potassium, because aldosterone is going to both kind of control potassium and sodium levels. And then also we know that electrolytes help maintain acid-base balance. And this is our ratio of hydrogen protons to bicarbonate. I'm not going to use the superscript if you're okay with that. There you go. HCO3, 3 should be small. Or subscript, excuse me. You know how to write that hopefully by now. So let's look at the roles of kind of the four big electrolytes that we've talked about at various times. Magnesium is the one we've talked the least about, but these other three we've definitely mentioned. All right, so let's highlight a few things and think about this for a minute. Sodium has a role in both fluid and electrolyte balance. It's the most abundant extracellular ion, and we saw that last lecture in that graph, how much it was. And we know it's essential for impulse transmission for both muscles and nerves, and it works with potassium doing that. And it's a key cation in fluid electrolyte balance. And this is because water follows sodium. So if there's sodium somewhere, then water will move with it. It also helps maintain acid-base balance by exchanging with hydrogen protons. So we can take a hydrogen proton and put it out, say, in our urine or somewhere else, and we can bring a sodium back, and that will balance out the charge and make the charge the same, but get rid of an acidic component like a hydrogen proton. Plasma sodium contributes to osmolality because we have fluid shifts. Remember, water follows sodium. Now, this is mostly review here. So I'm going to just write review. This is review from Bio 232. So we have aldosterone, and it obligatory reabsorbs sodium. So you might remember what that is. If not, you should go back and review. We have ADH, which is one of its good friends, and ADH decreases sodium concentration, or if there is decreased sodium concentration, we then have low ADH, and we have very dilute urine output. So if we have high ADH, we bring sodium back and water follows. Okay, and so ADH will determine whether we have dilute or concentrated urine. So in this case, we're showing dilute urine. So this is low ADH. Okay, and then we have ANP. And you might remember that ANP works opposite of aldosterone and ADH. So if we have a lot of ANP, we will try to get rid of sodium and water. Okay? And it inhibited the release of those two guys. Okay, so these two work together. We'll put a little note here. Remind ourselves they work in unison. And atrial natriuretic peptide inhibits those. Okay, so if you're not remembering a lot of that stuff, you probably want to go back and review it. And you can go look in both your, we talked a little bit about aldosterone and ADH also in endocrine this term, so you can go back and kind of review. Then I want to remind you also we have baroreceptors that are looking at blood volume, and if they pick up high levels of blood volume, one way they'll get rid of it is they will increase filtration rate, and they will get more sodium out and water will follow. All right, so here's the normal values for sodium, which I will underline. I don't know if I'd ask you specific numbers, but what I do want you to know is if you have too little or too much, what that means. And so too little, hyponatremia, we talked about that. That could be due to increased water intake. We could have really high ADH that keeps a lot of water coming back and diluting our sodium. We can have hypernatremia, where we are getting rid of water, but our sodium levels are staying high. And so that could be like diabetes insipidus, where we're making much high amounts of water. Okay. Now, potassium has a role also in fluid and electrolyte balance. One is that it's opposite of sodium, right? So it's our primary intracellular cation. So it's going to maintain fluid volume in cells, where sodium maintains fluid volume more in plasma. And it's going to shift in opposition of sodium ions. All right. And it actually shifts in opposite direction to hydrogen protons, so it maintains that charge balance, just like we were talking about above with sodium and hydrogen. So it will usually be opposite of sodium. It will usually be opposite of hydrogen ions. We haven't talked about acidosis and alkalosis yet, so let's wait on that one, and we'll talk about that later. I don't want to highlight that yet. And it obviously is also like sodium in that it's essential for impulse transmission of muscles and nerves. It helps maintain membrane potential and cell excitability. And it is regulated by aldosterone because sodium goes opposite of it. If we reabsorb sodium, we secrete potassium. Okay? So those switch. We have hypokalemia, and we have hyperkalemia. Those are our normal values right there. And so if we have low levels of potassium, sometimes we can get diarrhea forming because of water shifts. If we have hyperkalemia too much, we can sometimes we see this in burns. And the main reason why we see it in burns is because we have damaged muscle and skin, and so the potassium that's in those cells will come out and spill into fluids. Also, if we have renal failure and we're not getting rid of active potassium, that can be a problem. And we don't really talk about what high levels of potassium can do, but it can be cardiotoxic. So that's one thing to watch out for. I'm looking here in my little notes. Yeah, we can have cardiac arrhythmias. We have muscular weakness, and all sorts of problems with both nervous system and muscle, just like we did with sodium, whether we're at low levels or high levels. Okay. Now, calcium. Let's just say, well, let's first talk about this first part. So it's used in a bunch of different places. We kind of forget that calcium is not just in our bones, right? We use it for blood coagulation. It's used as neurotransmitter releaser for axon terminals. It obviously helps form bones and teeth, and most of calcium's there. But in the intracellular fluid, it's also used for muscle contraction and nerve contraction. So, or not nerve contraction, but let's just cross that out there. Nerve impulse continuation. Maybe that's better because it's dealing with the neurotransmitters. Continuation. There we go. Okay. So it's used in a lot of places. Neurotransmitters, muscles, our skeleton, our teeth, coagulation of blood. So it's all over the place. Now, I'm just going to say review for final what PTH does to increase levels of calcium in the blood. But you probably remember that we use bone, kidneys, and small intestines to have different things occurring that each one of those, and also it is going to help activate vitamin D, PTHs. So review what the functions of PTH are and how it works with calcium balance. And lab values, I think this is supposed to be 9.8. It's pretty tight control. I think that is an error. Let me double check my notes here really quick. Calcium. Oh, I don't have around 9. Oh, I guess it's around 9. So, okay, we can keep 8.8 is fine. And abnormal hypocalcemia, we sometimes see in acute pancreatitis. In hypercalcemia, we will have because of hyperparathyroidism, which we talked about when we talked about PTH. So you can review those. Magnesium. So this is the one that we haven't really talked much about. It's seen intracellularly as an electrolyte mainly. Okay. A lot of it is in the skeleton, and the rest is inside the cells. It is used for metabolism of proteins and carbohydrates, and also synthesis of those. So to make proteins and carbs, to synthesize them, we see magnesium show up. Low amounts of magnesium can lead to irritability and hyperexcitability of CNS. High level can depress the CNS, and because of some of the neuromuscular function that it has in terms of protein metabolism and synthesis. So it also can cause arrhythmias in the heart. What we usually do if we want to maintain magnesium is we will excrete it if we have high levels of calcium or magnesium. And that kind of makes sense because it's involved in skeleton so much. If we have a lot of calcium, we usually have a lot of magnesium, and we may want to excrete it. Okay, so that's one way we can get rid of excess. If we see too much magnesium, we'll excrete some. If we see too much calcium, often magnesium's high too, we'll excrete magnesium. Low levels, as we said here, we already talked about this. What causes low levels? Well, often nutritional deficiency is the cause for low levels. High levels is usually due to renal failure. Okay, so and we've already mentioned what those things can cause here, the hypo and hypermagnesia. That's really all you need to know about magnesium. It's not one that we cover in a lot of detail, so that part is fine. What I want a little more time spent on is acid-base balance. And so what we're going to spend the last 4 or 5 pages on here is going over acid-base balance and kind of understanding it a little bit. And so we have a pH balance. And we know that our blood is very tightly controlled. It's around 735 to 745. Arterial blood tends to be a little bit more basic. the venous blood a little bit more acidic because of waste products coming from the tissues. The lower the pH is due to those acidic metabolites coming from tissues. So things like high carbon dioxide, high lactic acid, high hydrogen protons. Okay. So we're in alkalosis if we're above 7.45. We're in acidosis if we're below. And we really can't go super far away from this. If we go very far away from the 735, 745, we can actually get really sick really fast and can go into comas and die. Most of these hydrogen protons are produced via metabolism. They're end products of metabolism, and we need to process them, right? We want to get rid of these as quickly as we can. And we have 3 different systems to get rid of excess hydrogen. Okay, we have chemical buffers, we have respiratory center, and we have the renal mechanism. Okay, so we're going to go over those areas in a little detail and talk also about the different chemical buffers we have. Okay, so we have chemical buffers that act in milliseconds, and we have our 20-to-1 ratio of bicarb to hydrogen protons in plasma. That's one of them. We'll talk about some of the other buffers in a minute. We have a respiratory center, which acts in minutes and adjusts our breathing rate up or down. And we're going to also talk about how that works in a moment. And then we have our renal mechanism. And this is the one that can remove hydrogen protons from our system by putting them in urine, and this can act in hours to days, and also the renin-angiotensin mechanisms involved in this as well, because we're going to change how we bring water back and what leaves and comes into the kidney. So let's now talk about the chemical buffers first. There are 3 chemical buffers. There are 3 systems, right? So we also have 3 here. So we have 1. Excuse me. It's been a great while. It likes to do that. Okay, so we have 1 right there, and then we have 2 right here, and then we have our 3rd one right here. Okay, so bicarb buffer system. All right, this is the one that, oh, and so by the way, these pictures, so if you go to these pages, these pictures are in detail here, page 1030. And you can read about them there if you want. Oh boy, something's gone wrong. Hold on. Let's start over. Let's try that again. Page 1030. Page 1031. And yes, you guessed it. Page 1032. Okay, for each of these 3. Now, what I want you to focus on is not memorize these drawings over here. But what I want you to think about is a couple different things. 1, the name of the system. So in this case, the bicarb buffer system. 2, where it's buffering. So it buffers blood and interstitial fluid. And blood is specifically the plasma, right? When we say blood, we're really, in this case, referring to, I can't put it in here, but we'll put it here. Blood, plasma, right? When I say buffering blood, the fluid portion. And what it does is what we've kind of already talked about, where we have what's basically happening is we have this large amount of bicarb ions outnumbering carbonic acid or hydrogen protons at about 20-to-1, and we need to maintain that, right? This is something that we want to maintain. We want to, every time a hydrogen proton is produced from metabolism, we want to have a bicarb that can go ahead and can sequester it. And so what we're seeing over here is we see bicarb, all right, going with a hydrogen, making carbonic acid, and then we will take the water, and the water will go out in our urine, right? Go out to urine, And then we have carbon dioxide, and the carbon dioxide will go out. Why don't we circle it in a different circle here? Carbon dioxide, and carbon dioxide will go out. Oops. Go out to lungs, right? So that's how we get rid of our things. And then what we want to think about is if we used one of these bicarbs, our ratio is going to drop, right? Every time we use one of these, we're going to lose stuff over here. We're also going to lose stuff here, but remember when we talked about this ratio in 232, the problem is that every time we add a hydrogen proton to the ratio, it's so much, because there's so much less of it, it makes such a bigger deal. It's so much more important. And so we want to keep this 21 ratio all the time. So without going into all of this detail, I just want to show you that we used a bicarb here. We bring back a bicarb, all right? And so as a result, there's the one that we bring back. As a result, we conserve the 20-to-1 ratio. Okay? So in this particular buffer system, if everything's working correctly, we can buffer our blood and our interstitial fluid by grabbing onto excess hydrogen and getting rid of the water in our urine and getting carbon dioxide breathed out. And we will go through other processes to go ahead and restore the bicarb balance and keep the 21 ratio. So even though we use a bicarb, we can remake it in effect and conserve it. Now, the phosphate buffer system is a little bit different. So the phosphate buffer system uses phosphate buffers. These are buffers that have phosphate groups in them, and they're very useful for buffering filtrate and urine as opposed to plasma and intercellular fluid. And so in this case, because we use a different buffer and we get at hydrogen and we have it go out in our urine, okay, so we capture that hydrogen and remove it, what we do is we're making a new bicarbonate, okay? So we make new HCO3 negative, right? And that's important because that will help us keep that 20-to-1 ratio when we have a lot of acidic metabolites. So we always want to be making more of this bicarbonate. So in this particular one, because we're using a different buffer in our urine system, we actually, by breaking that hydrogen off and getting rid of it, we make bicarbonate and it goes there and it's new. Now notice in each case there's a chloride shift that's occurring. Remember our chloride shift from 232? So we take a negatively charged ion and bring it outside, and we bring a negatively charged chloride in. So just as a little note there, that's still happening. Our third one is the protein buffer system. Now in the protein buffer system, we tend to see this in body cells and blood plasma, right? And this is coming from the amino acids, and amino acids are amphoteric, which means that they can either act as an acid or a base. And this is because they have a carboxyl group, which you might remember from your intro bio, and an amine group. And the carboxyl group can donate hydrogen ions, and the amine group can accept hydrogen ions. And so we can take an amino acid, they're using glutamine here as an example, and we can break it apart into these groups. And then what we can do is we can get rid of our, we can grab on the hydrogens and then get rid of them as an ammonia complexes. So see all the hydrogens that are in there, and those go out into the urine. Okay. Okay, so let's circle it so we can see what we're writing. So, but notice what happens when we do that. We can take and turn this carboxyl group into bicarbonate, and we can make new bicarbonate. In this case, it even makes two. Make new HCO3 negative, right? So those two this one at top here maintains the ratio as best it can. It basically, for using one, we kind of get one, and that's good. These two make new bicarbonate to make sure that ratio stays at a 20-to-1 rate, even when we make a lot of acidic metabolites. So this is good. So these buffer systems help each other make bicarbonate that'll be in this first buffering system in our blood. Because remember what we said, we really want to keep our pH right around 735, 745. We need to do that, otherwise we can get really, really sick. Now, along these lines, we want to now talk about the respiratory system and how it goes ahead and regulates ion concentrations. And we need to look at our equation that we looked at a little bit in 232, which is this carbonic acid bicarbonate reaction. So what happens here is when we have carbon dioxide somewhere, we can combine it with water and it can form carbonic acid. Carbonic acid then can dissociate into hydrogen, proton, and bicarbonate. Okay, so this bicarbonate is going to be used to grab onto these if we have too many. So we can run the equation this direction as well. So if we have a lot of hydrogen protons, we can use a bicarb, make carbonic acid, and then turn it into water and CO2. So depending on which way we run the reaction, different things will happen. So let's say, okay, so let's think about our respiratory system, because respiratory system is going to move carbon dioxide in and out of our body, right? So let's say we have really fast breathing, hyperventilation. Okay, so if we hyperventilate, the amount of carbon dioxide in the body fluids is going to go down because we're going to increase a lot of CO2. Okay, so that's going to mean that this equation is going to run in this direction. So I'm going to put an arrow over here. The equation's going to run that way. Okay, that's the way the equation's going to move, is this direction. Okay, that means that we're going to grab a lot of this hydrogen proton and get rid of it by breathing out the carbon dioxide. So the pH should go up and we should get into an alkalosis state. It's going to pull the reaction to the left. Okay, it's going to pull. Here, you want me to put the arrow over here since that's where we're talking about it? Maybe that makes more sense. Let's draw a better arrow in here. We're going to pull the arrow to, or the reaction to the left. Okay. All right. Now, what if we're breathing slowly? All right. Slowly, we'll hold on to carbon dioxide in the tissues, so less carbon dioxide gets exhaled and stays in the body fluids, and therefore, here, let's do it this way. Less gets exhaled, more stays in the body fluids. So the pH will go down because more of this will stay in the body fluids. We'll combine it with water. It will turn into this, and then we'll get extra hydrogen protons and bicarbonate. Remember, when we have a 20-to-1, when we have a 20-to-1 ratio, this is what I was talking about a couple minutes ago. When we have a 1-to-20 ratio of these, if I add one more of these and I add one more of these, the ratio actually goes from 20-to-1 to like 21/2, and so the ratio is dropped. So every time we add one over here and we add one here, the ratio actually switches, and that can be problematic. And that means that whenever we make this, the ratio gets out of balance and we get more acidic. All right, and we're going to go into acidosis when this happens. All right, we're going to be in acidosis. That's going to push the reaction to the right, and if it pushes the reaction to the right, we will have more acidity. Okay, so our respiratory system can change whether we're going to have a pH in our blood plasma that is acidic by breathing slowly or holding on to more carbon dioxide, and our faster breathing can push more CO2 out. Now, this usually is all regulated by the respiratory center, the medulla and the pons, and it's going to notice what's going on in terms of how much acid we have in our blood, and will increase breathing or decrease breathing. So think about when you go for a run or do some exercises, your breathing rate increases, you hyperventilate, because you're producing a lot of CO2 and we need to exhale it, so we need to run the reaction towards the left to get rid of more of this, right? Because once it's gone, the reaction can't go backwards because the CO2 is gone. And pH changes of about 0.2 up or down can double or halve the breathing rate. So that's significant, right? Just a very small amount here, pH changes can really double or halve the breathing rate. So that's kind of a lot. Now, we have then respiratory acidosis or respiratory alkalosis. And this is what we're talking about here. We're either going to do something. So the way to think about this is if we see the word respiratory in front, we know something's happening to the lungs, and the lungs are either not getting out CO2 or getting out too much CO2. Okay? We also have metabolic acidosis and alkalosis, and this is due to not the lungs being involved, but other things in our body, some metabolic processes. So we'll get to those in a minute. But let's look at these two. Okay, respiratory acidosis. All right, so this is going to be where we're going to be holding on to CO2 in our blood and not getting it out. So this can result from different things. Head and chest trauma, but especially from respiratory pathology or diseases. Okay, diseases. For some reason, our lungs are not working correctly, and they're not getting the carbon dioxide out of the blood fast enough. So what could be some of these problems? Well, emphysema, pneumonia, edema, fluid in the lungs, injury, and then as we said up here, head trauma, injury to the respiratory center of the medulla. All of these cases, the lungs do not expel enough carbon dioxide. Okay? Now, how would we treat it? Well, we could treat it with bronchodilators and small amounts of oxygen to increase air exchange in the lungs. So we could treat it that way. And obviously, anything that interferes with breathing is going to cause this accumulation of CO2. So any asphyxia that occurs can cause this. All right. So that's respiratory acidosis. Our body's holding on to carbon dioxide because we cannot breathe it out fast enough. Respiratory alkalosis is the opposite. The body is being stressed and it's going to be expelling more CO2 than it necessarily needs to. So the body's being stressed, maybe by shock, by some sort of sepsis or infection, some sort of trauma, or even sometimes in the case of asthma. Also, high altitudes or severe anxiety can change our breathing rate. And if we breathe faster and actually expel more CO2 to get more oxygen, but we expel too much CO2, we can get into respiratory alkalosis, because remember, we're going to run the equation to the left if we do that, and we're going to take out so much of this acid here that we're going to actually, our pH is going to go up instead of down or stay the same. So in hyperventilation, we're going to eliminate an excessive amount of CO2 because too much acid gets blown off, right? And there's a little bit of description here about the feelings that sometimes we experience when that's happening. Okay, so know the difference between respiratory acidosis, respiratory alkalosis. And then you need to contrast that with metabolic acidosis and metabolic alkalosis. So metabolic acidosis results usually from abnormal amounts of acidic metabolites other than carbon dioxide, okay? So this is usually due to high amounts of acidic metabolites that aren't carbon dioxide. And these can be caused by severe diarrhea, by renal disease, not getting rid of acidic metabolites, untreated diabetes mellitus, especially type 1, starvation. We can also not have enough bicarb, right? If we don't have enough bicarb, then we can't, if we're low on bicarb here, we can't grab onto this and run the equation in that direction, so this accumulates, all right? So the other things where we said we have lots of abnormal acidic metabolites is we have so many acidic metabolites, we're overwhelming our bicarb. And so that's what happens in metabolic acidosis. Or any increase in acid production. Any of those things high levels of metabolites that are not carbon dioxide, but other things that we're producing cause metabolic acidosis. So think of other things that will cause this: drug intoxication or abuse, severe illness, right, where we aren't getting rid of our acidic metabolites, high amounts of alcohol, high amounts of lactic acid, high amounts of keto acids and ketones. All of these things increase the acid in body fluids, and that lowers blood pH. The low levels of bicarb can be coming from excessive diarrhea because we're getting rid of too much bicarb, or our renal tubes are excreting too much bicarb. So that could happen too. That's a possibility. So here's a very simple short-term, like when your leg falls asleep, you're going through a metabolic acidosis because you're blocking blood flow and you're getting acidic metabolites accumulating in an area. That's very minor. These other ones that we've underlined are much more severe. Now, metabolic alkalosis is a lot rarer. Sorry that these thes are not where they're supposed to be. I don't know what happened there. This is a rare condition. Okay, but so, and the main reason why is we almost always make acidic metabolites, not alkaline metabolites. So we don't often have our blood pH go above 7.45. But if we did, it might be due to increased plasma bicarbonate levels. So that could be due to excessive vomiting. Why? Well, we're going to lose a lot of hydrochloric acid. And so as we make more of it, that's going to actually make the amount of bicarb look at a higher ratio because we're taking out so much hydrogen protons from the blood. So that kind of shifts the equation a little bit. We could ingest a lot of alkaline drugs such as antacids and overdose on things. And so if the body becomes too alkaline, the nervous system and heart can be affected, obviously, because we're going to have a shift in our electrolytes. So this one, much less common, but still worth knowing. So anything, and as I said this earlier, anything that is not very far away from 73545 is really bad for us. And so we usually will say anything below 7 is acidosis, and we usually have depression of CNS, and the patient will usually enter a coma and die if it's prolonged. And usually above this, we overexcite the CNS, and this can also lead to convulsions, tetany, our muscles not working well, and ultimately death. So we want to keep our pH of our blood in very tight control, and that's why we use all these different mechanisms we've been talking about to do that. Okay, one last thing, and a little bit of this is review. When we looked at diabetes, and a little bit of it is new. So I'm going to add a couple things in here. So we want to talk about fatty acids and beta oxidation. So if you increase your fat intake, or if you're undergoing starvation and your body starts to use your body fat more as its nutrient source, we're going to have a large amount of fatty acids in the bloodstream and ketone bodies. Okay, now we'll talk about those ketone bodies in a second here. So this is normal. So this diagram right here is the normal route for fats. Let's do the normal metabolic route for fats, okay, for lipids. And so what happens is we take our lipids and we break them into our monomers, our glycerol and our fatty acids, right? And then the fatty acids undergo a bunch of things here, and they're beta oxidized, and they eventually become acetyl coenzyme A, and they enter the Krebs cycle. So kind of similar to what glucose does. And the glycerol gets converted into pyruvic acid, and it eventually enters the Krebs cycle as acetyl coenzyme A. So that can happen. So we can use fat as a primary energy source, and that's okay. All right. Now, when we have a lot of fats and fatty acids, we get a lot of acidity in our system, and we get what's called ketosis. And prolonged ketosis can cause metabolic acidosis, because remember, metabolic acidosis is high levels of any metabolite that's not carbon dioxide that's in our blood. So we're going to start with what happens normally. Normal. So the first step normally is that fatty acids are broken down into a 2-carbon acetic acid fragment, and they're fused to coenzyme A, and they become acetyl coenzyme A, right? And that's where they enter the Krebs cycle. They then enter the Krebs cycle normally. Nothing unusual here. Now, the second step, the acetyl coenzyme A enters and produces ATP, right? We go through our normal aerobic respiration. And this happens, this acetyl coenzyme A enters and produces ATP, and it happens via this thing called oxaloacetic acid. And this picks up the acetyl coenzyme A and takes it to the Krebs cycle. Okay? Now, so we're going to put, but if there are too many fatty acids because of what we said up here, too many fatty acids due to maybe increased fat intake or starvation, but if there are too many fatty acids, then there becomes insufficient oxaloacetic acid, right? Because if we have too many fatty acids, we don't have enough of this transport. So then what happens is this these fatty acids become acetoacetic acid, and they get converted into acetone, and the acetone ends up in the blood, and we have that acidosis. And remember, some of these acetone can be liberated in our exhalation, and that was what we investigated when we were talking about diabetes, type 1 especially, and the acetones going out. So if the person's out of control, insulin control, and they aren't getting enough nutrients, their body switches over and uses a lot of fatty acids. If they use a lot of fatty acids, they have too many fatty acids in their bloodstream, we can go ahead, we get ketosis, we get this problem where we don't have enough of the oxaloacetic acid, so the acetyl coenzyme A forms this substance called acetoacetic acid, and that gets converted into acetone, and we get, unfortunately, acidosis. So people who have type 1 diabetes that's very uncontrolled for long periods of time, they definitely can get metabolic acidosis, and it can cause them lots and lots of problems. All right, so last thing, we have a 2-page sheet here. This one is blank. This one has the 4 different acidosis: respiratory acidosis and alkalosis, metabolic acidosis and alkalosis. And then what it does is it gives you the ability to kind of fill in some things here if you want about the different concepts. And if we look here, here are the answers. So you can kind of figure out what you're supposed to put in for each, or you can read this and kind of study it and then see if you can do that. Now, I'm not going to ask on the exam for you to fill in a chart like this, but this chart can be a good way to study and a good way for you to review material. So if you want to, take all those pieces on the last couple pages, put them together into that chart, and that might be a good way to study for the exam. So, all right, we're done with fluids, electrolytes, and acid-base balance. And if you have any questions, talk to your instructors, and we will chat later. Bye.